So welcome to the next HL topic, nucleic acids. By the end of the lesson, you should know that nucleotides are condensation products, that polynucleotides are also condensation products, that what DNA and RNA are, the sequence of bases in DNA and what it determines, and we'll have a focus around GMOs. You should also be able to explain the stability of DNA in terms of the interactions between hydrophilic and hydrophobic components, explain the origin of the negative charge of DNA, deduce the nucleotide sequence in the complementary strand of DNA, and explain how the complementary pairings between bases enable DNA to replicate itself exactly. And finally, discussing the benefits and concerns using GMOs. So first, nucleic acids. So nucleic acids are condensation polymers of nucleotides, which in turn are the products of the condensation of a nitrogenous base, a pentose sugar, either ribose or deoxyribose, and a phosphoric acid. In order to understand the functions of nucleic acids in living organisms, we need to discuss first the structures and properties of their components, the nitrogen bases, and the nucleotides. When nucleotides combine with one another, the phosphate groups form diester bridges between the 3' and 5' carbon atoms. Okay, we see them here, of adjacent pentose residues. So when nucleotides combine with one another, the phosphate groups form diester bridges, as we've previously looked at, between the 3' and 5' carbon atoms of adjacent pentose residues. Now, further condensation reactions produce long polypeptide chains known as strands, in which monomeric units are joined together in strict order and orientation. Now, just like proteins, each DNA and RNA strand has two terminals. Okay, so you've got the 5' end and a 3' end. In living cells, the synthesis of nucleic acids begins at their 5' end, and so the sequence of nucleotides is traditionally recorded in the same way. So for example, for this sequence, it would be GTACA, GTCA, CC. Now, DNA molecules consist of two polynucleotide strands in which each nitrogenous base from one strand forms a complementary pair with a nitrogen base from another. Okay, so we see that here. Each pair contains one purine base, an A or a G, and one pyrimidine base, a T or a C. 2 hydrogen bonds at the AT base pairs and 3 hydrogen bonds with the GC base pairs, as shown. The double helix shape of the DNA molecule stabilized by hydrogen bonds between the complementary nitrogen bases is known as its secondary structure. Okay, that's the famous double helix. So essentially what happens is that anionic and nucleic acids wrap around the basic positively charged proteins known as histones. So in our body, physiological pH, it's slightly alkaline. So the outer phosphate groups of the DNA strands ionize to form basically a large anion. This is hydrophilic and forms hydrogen bonds with water. The hydrophobic and hydrophilic parts of this polynucleotide chain stabilize the DNA helix and allow it to become resistant to chemical cleavage. So in terms of the nitrogen bases, nitrogenous bases are heterocyclic aromatic amines that contain several nitrogen atoms and act as a proton acceptor in aqueous solutions. All common nitrogenous bases are derived from two parent amines, the pyrimidine, okay, which is this kind of system here, and the purines. The pyrimidine nitrogenous bases are simply pyrimidines, include cytosine, thymine, and uracil. And the purine nitrogenous bases commonly are called purines and include adenine and guanine. The names of the pyrimidines and the purines are often abbreviated to their first letter. So, for example, adenine is A. Okay. So essentially, thymine is normally associated with deoxyribose sugars and is found in DNA, whereas uracil, U, forms nucleotides with ribose and is found in nucleic acids R and E. Therefore, both DNA and RNA contain 4 nitrogenous bases each, including 2 purines and 2 pyrimidines. The pairs adenine and thymine, and adenine and uracil, and guanine, cytosine, and guanine and cytosine are known as complementary base pairs. This ability of certain nitrogen bases to form hydrogen bonds with one another in specific order and orientation plays an important role in sorting out processing information. So, in contrast to amino acids, nitrogenous bases are almost insoluble in water because their molecules are held together by strong hydrogen bonds. Thymine or uracil can form 2 hydrogen bonds, whereas with adenine, while cytosine and guanine bind with each other with 3 hydrogen bonds, which is what you see here. So, nucleotides form by making linkages with nitrogen of a base and the ribose sugar group, and we've already looked at this. The nucleotide consists of a phosphate group, a pentose, and a base, and this occurs during condensation reactions. The nucleotides start off being monomers, and through the condensation reactions begin to form this nucleic acid polymer, which creates the chain. And as we've discussed, nucleotides link up with hydrogen bonding between oxygen and carboxyl groups and hydrogens of amine groups. So again, adenine forms 2 hydrogen bonds with thymine, and guanine has 3 hydrogen bonds with cytosine. 3 single hydrogen bonds with cytosine. So in terms of summarizing DNA versus RNA polymers, so uracil replaces thymine and RNA, and also the nature of the bases in terms of the ribose sugar are different in RNA and DNA. So in terms of DNA synthesis, which is also key, okay, it's broken down into 2 processes. Okay, transcription comes first. So essentially what happens is that first group of enzymes called initiator proteins separate the 2 DNA strands and create a short polypeptide fragment known as a primer. And this is paired with the secondary strands by complementary nitrogen bases. Another group of enzymes known as DNA polymerases add more nucleotides to the primers using the existing DNA strands as templates. The resulting new polynucleotide chains are complementary to existing DNA strands and therefore produce 2 identical copies of the original DNA molecule. Finally, the replication process is terminated either by a certain sequence of DNA or by the action of proteins that bind to a specific DNA region. And then we move on to transcription. Okay, so this involves a RNA molecule onto a DNA template. So during transcription, a DNA sequence is read by the RNA polymerase, which produces the RNA molecule complementary to an existing DNA strand. In contrast to the original DNA, the resulting RNA molecule contains a ribose sugar instead of the deoxyribose of DNA and uracil nitrogen base instead of thymine. In addition, RNA molecules usually exist in a single polypeptide strand with various 3-dimensional configurations. The exact shape of an individual RNA molecule is known as its secondary structure and is determined by the hydrogen bonds between complementary nitrogen bases from different regions of the same strand, just as DNA has that double helix secondary structure. Each type of nucleic acid plays its own role in hereditary. DNA residues in chromosomes store genetic information and act as a template in which can be copied onto the RNA. The resulting RNA molecules transfer the genetic information from chromosomes to other regions of the cell and in turn are used as templates for protein synthesis. This is known as translation and occurs in ribosomes. All living organisms use the same genetic code, and that allows ribosomes to translate 3 nucleotide sequences known as codons into sequences of amino acid residues on polypeptide chains. Okay, so basically the RNA gets transformed into a particular amino acid. So just to finish off with GMOs, so a detailed understanding of DNA structure and function leads to the development of DNA manipulation. These techniques are known as genetic engineering and allow scientists to alter DNA sequences. This includes the transfer of genetic material from different species. This results in something known as a GMO or a genetically modified organism. Most common GMOs are in transgenic plants, for example, BT corn, which contains a gene from a bacterium that basically creates a toxin that destroys the common pest of that particular plant. And because of the effectiveness of this GMO, it's now become the predominant corn in the world. There are several benefits to GMO. You can lead to longer shelf life, improve flavour, increase resistance to diseases and pests, as we saw with the BT corn. It can produce a supply of vitamins and vaccines, for example, the vitamin A rice in India, increasing crop yields. And you can have a tolerance for wide growing conditions, for example, making them more resistant to drought. So you can grow the plants during times of drought. Of course, there are always concerns, and these have included the lack of information about long-term effects, possible link to allergies in foods, changing ecosystems, altering the composition of food, and concerns about breeding species that are resistant to control, as they reproduce, and finally a lack of information about food labeling.